Assalamu alaikum. Let's do DNA replication now. So, um, so a genome is basically the entire content of a cell. Okay, so the all 46 chromosomes in our somatic cells are known as a whole a genome, and DNA is organized as chromosomes in our cells. You already know that. Now in bacteria there is only one chromosome and it is circular. However, in eukaryotic organisms like yeast and human, um, DNA is organized as linear chromosomes, and they are complex with histones. Okay, so the thing is, the scientists didn't know how chromosomes are replicated because this is necessary for continuation of life. And they came up with three theories, and they said that basically you have two original chromosomes in black, and you have two daughter cells, okay? And these daughter cells, each one of them would have the two original chromosomes, and one would have two new chromosomes. And they thought that it was dispersive. That is, you have portions of the parental chromosomes and portions of new chromosomes in the same chromosome. But these theories turned out to be wrong. So scientists knew by doing a few experiments that it's actually DNA is replicated in a semi-conservative fashion, meaning that um, one strand is conserved. Okay, so semi means sort of partial uh, uh, conservation of DNA. So one of the chromosomes comes from the parental cell, and one is actually a whole new chromosome. So how does that happen? Well, it turns out by doing again more experiments that DNA is replicated in a bidirectional manner, meaning that it starts at a single point and it continues in two opposite directions. And this is how DNA, the new DNA, is labeled. Okay, so you can see how there is sort of like a bubble that looks something like this. And they said, it starts right here at this point, and then it goes into opposite directions. Now, the thing is, when DNA is replicated, DNA synthesis always goes from the 5' end to the 3' end. Okay, so right here you have the first nucleotide, and the incoming nucleotide, the new nucleotide, is added to the 3' end of the existing nucleotide. Okay, so whenever we want to add a third nucleotide, we add it to the 3' end of the second nucleotide, and so on. Okay, now remember that DNA is anti-parallel, so the template strand is read from the 3' end to the 5' end. Okay, so remember that. So we have a replication bubble right here. So let's focus on one half of this bubble. Okay. So it turns out that DNA is synthesized in two different manners on both sides, on both strands. So in one strand, the new strand is synthesized continuously. So, and it goes from 5' to 3'. Whereas on the opposite strand, DNA is synthesized discontinuously. So it is synthesized as short fragments. And these short fragments are known as Okazaki fragments. So scientists thought that DNA would be synthesized continuously as well, going in this direction. But it's against chemistry, right? Why? Because in this case, it would go in the direction of 3' to 5', and the template would be read from 5' to 3'. But this is not true. So in order for DNA to be synthesized in the direction of 5' to 3', it is synthesized as short fragments. Eventually, these short fragments would be connected to each other, forming one continuous long strand and it is known as the lagging strand. So why is it known as the lagging strand? Because it is lagging behind, meaning that it is not finished until the other strand that is synthesized continuously is finished. And this strand is known as the leading strand because it's leading the way. So what it's doing actually is that as it is synthesized, it opens up the fork further, allowing for a new short fragment to be synthesized. Okay, again, these short fragments are known as Okazaki fragments, um, based on the name of the discoverer of this feature, of this phenomenon. Okay, so you have a leading strand and you have a lagging strand, and the lagging strand waits for the leading strand to open up the fork further to allow for the synthesis of another Okazaki fragment. So in this case, DNA is synthesized in the direction of 5' to 3' for both strands. Okay, so what I want you to do actually is, this is only one half of the bubble. What I want you to do is to draw the other half of the replication bubble and see how it looks like. See how DNA is synthesized. So this is a homework for you. So DNA replication is really a complex process, and it requires a number of molecules to complete it. And we'll talk about these components right now, the molecular components. So the first thing that is needed is actually a primer, meaning that something to start replication. Okay, and it turns out that DNA polymerase cannot start synthesis de novo. De novo means from scratch, okay? That is from nothing. It needs something to allow it to start DNA replication, and this thing turns out to be an RNA primer. So an RNA primer is synthesized by an enzyme known as a primase. So this is synthesized again in the direction of 5' to 3', and then the DNA polymerase starts from the end of the primer and it continues DNA synthesis. So each leading strand requires one primer, right? Because you need this primer and the leading strand just keeps on synthesizing DNA. On the other hand, each Okazaki fragment requires a primer of its own. So we have DNA synthesis going in the direction of 5' to 3'. That's the leading strand, this one right here. Okay, but we have an Okazaki fragment, and then we have an RNA primer to start the synthesis of another Okazaki fragment. Okay, now, so what happens then is that the DNA polymerase keeps on synthesizing the Okazaki fragment until it hits the primer. Then it falls off, it stops. And another DNA polymerase comes in, and what it does is that it removes the primer and it continues to synthesize DNA in its place. And then you have a DNA ligase to connect the two Okazaki fragments together. Now pay attention to this. So we have a DNA polymerase, and in bacteria it is known as DNA polymerase 3. It synthesizes each Okazaki fragment. But then when it falls off, you have another one, and it's known as DNA polymerase 1. What it does is that it removes the primer and it replaces the space, empty space, by DNA, deoxynucleotides. Now, and it's known as DNA polymerase 1 again. Okay, so removal of the primer requires an enzymatic activity known as an exonuclease. Okay, so a nuclease is an enzyme that degrades nucleic acids. Specifically, in this case, it's DNA. So what DNA polymerase 1 has is an exonuclease activity. So it degrades the, in this case, and this exonuclease activity is specific for RNA, because it removes the RNA primer. Okay, and then you have the DNA polymerase 1 filling in the gap. Okay, so this DNA polymerase 1 has an exonuclease activity. Now notice, in addition to its polymerase activity, now notice something that's really important, is that the direction of the exonuclease activity is 5' to 3'. Okay, so meaning it goes in this direction right here. So it starts right here and goes in this direction. So it goes from the 5' end to the 3' end. So until it hits this DNA, the other okazaki fragment, and it falls off, and a DNA ligase comes in and it connects the two fragments together. Now we do need other proteins as well, and these proteins are a helicase, and we need single-strand DNA binding proteins. So why are these needed? A DNA helicase is important for separating the two strands from each other, okay? To allow the DNA polymerase to do DNA synthesis. How about the single-strand DNA binding proteins? These are also known as replication protein A, okay? And okay, but first, DNA helicase separates the two strands from each other. It requires ATP and it's connected with the primase in a complex known as primosome. Now, remember that this is something that is important for proteins and enzymes, is that they tend to form complexes in order to process any mechanism really fast. Okay? So we have single-strand DNA binding proteins, and these are really important for three purposes. The first thing is that they prevent the formation of short hairpin structures. Okay. Hairpin. Pin means dabous, hair, شعر. So it's basically مثل البكلة. So what it does basically is that sometimes you can have within the same strand complementary sequences that tend to form a hairpin. Now, what hairpin structures do is that when the DNA polymerase goes on to synthesize DNA, these hairpin structures block the movement of the DNA polymerase. So what single-strand DNA binding proteins or the replication protein A does is that it prevents the formation of these hairpin structures, so DNA polymerase continues with the DNA synthesis. Now the other thing is that cells do not like to see single-stranded DNA. So what they do is that they tend to degrade it. So these single-strand DNA binding proteins, they prevent the degradation of DNA. Something else is that they stabilize the formation of the single-stranded structure of DNA, because remember, we have two strands that are complementary to each other. So what these proteins do is that they prevent the reformation of double-stranded DNA because they're complementary to each other. Okay, so in prokaryotes, there are three DNA polymerases. We have DNA polymerase 1, we have DNA polymerase 3, and we have we have 5 DNA polymerases, 1, 2, 3, 4, and 5. Now, number 3 is the DNA polymerase that is synthesis for the formation of the newly synthesized DNA. And usually it is a complex with the primosome, which is made of the helicase and the single-strand DNA binding protein, and it forms with this complex what is known as a replisome. Now, DNA polymerase 1 is important for DNA synthesis, all right, and it also has an exonuclease activity. As I said, it removes the primer. Okay, and number 3 is that, as we will learn in next lecture, it's also important in DNA repair. Now, the other DNA polymerases 2, 4, and 5 are important for DNA repair as well. Now, let's look at DNA polymerase 3. This is really a huge protein. It is not a simple protein. And this protein contains many subunits: alpha, beta, gamma, delta, etc. And what's important about these subunits, is that the alpha subunit is the subunit responsible for DNA synthesis. Okay, but there is epsilon subunit, and it has an exonuclease activity. Okay, and this exonuclease activity is different than the one that DNA polymerase 1 has, because it goes in the opposite direction. It goes from the 3' to the 5' end. So what does it do exactly? It makes sure that DNA synthesis is accurate. So even though it is not really highly probable, but sometimes DNA polymerase 3 makes mistakes in placing the right nucleotide. So what it does sometimes is that it sees a T and it adds an A. It sees another T and instead of adding an A, it adds a G. So yes, things happen. So DNA polymerase 3 uses the exonuclease activity to go backward. So it goes in the direction of 3' back to 5'. It removes the wrong nucleotide and it places the right one instead of it. Okay? So it makes sure that that DNA replication has high fidelity. Fidelity means ikhlas. Okay, so that it's really accurate. Okay, so but we have a problem, and the problem is that the DNA helicase separates the two strands from each other. But what happens is that DNA ahead of the DNA polymerase, which is right here, becomes overwound. Okay, it's almost like doing this act right here. You have overwound DNA, which makes it impossible for DNA polymerase to continue with the DNA synthesis. So how is this solved? It is solved by enzymes known as DNA topoisomerases. Okay, and there are two enzymes, topoisomerase 1 and topoisomerase 2. Topoisomerase 1 produces a transient single-strand break, so it sort of like creates a swivel. هذا هو السwivel, meaning that it separates the two chains or two strands from each other. So when DNA is rotated, only one portion of it is rotated and this is not rotated whatsoever. So that prevents the formation of the overwound structure. Okay, so here we have a look at how DNA is replicated. Okay, now, so we have right here we have the primase, and the primase is associated with the helicase, forming the structure of a primosome, right? And this structure can also be complex with the DNA polymerase 3, okay, and forming the replisome. Now, what happens is that you also have DNA polymerase 1. DNA polymerase 1, here you have an Okazaki fragment. What DNA polymerase 1 does is that it removes the primer right here between this newly synthesized DNA and the other DNA ahead of it, the other Okazaki fragment ahead of it, and it fills in the gap, and DNA ligase then comes in and it connects the two strands from each other, okay, with each other. Now, you also have ahead of the replication fork, you have the topoisomerase 1. Now, there's another enzyme known as topoisomerase 2, and we will talk about it in a second. Now, it turns out that DNA replication in bacteria does not start at any exact point. It rather starts at a certain stretch of DNA sequences known as an origin of replication. And this origin of replication is actually a consensus sequence. So it's one of those many consensus sequences that exist in bacterial DNA. So what do we mean by a consensus sequence? A consensus sequence is a sequence that is agreed upon, meaning that you can find the same sequence in different bacterial species and strains. Okay? So it is agreed upon. Okay? Like in the parliament, for example, when they take a decision unanimously, بالإجماع, we say that there is a consensus in terms of the decision. Okay, so consensus sequences that exist in the origin of replication are of two types. You have a 9 base pair region and you have a 13 base pair region as well. Now, the 13 base pair region is actually a tandem array of sequences. Okay, so you have the same sequence right here, right there, and right here as well. And these sequences are AT rich. Now, the other sequence is right here in blue. They are scattered, but within the same region as well, but it's not one after the other. So they are not tandemly organized. Now, the DNA, the 9-mer is actually the binding site of a protein known as DNA-A. Okay, and what DNA does is that it binds to the 9-mers, it forms a structure like here, and it squeezes on DNA, opening up the AT-rich sequences. So now we know why these sequences are AT-rich, so that the interaction between the two strands becomes weak because these sequences are AT-rich. Okay? So that's what DNA-A does. Now, when cells see a bubble, they know that it's time to replicate DNA. So the helicase and the single-strand DNA-binding proteins, they jump on DNA and they start opening up DNA even further. And DNA polymerase 1 binds, DNA polymerase 3 binds to the replication bubble, and it starts with the DNA synthesis. Now, what happens in bacteria is that synthesis is bidirectional, so it goes in both directions, and until it hits the other side of the DNA of the chromosome, and then the two strands are separated from each other. Now, notice that you have one new strand and one old strand at the end of DNA replication. Well, how about in humans? What does happen in the human genome? The human genome is huge, so one origin of replication is not enough, and as a result, there are multiple origins of replication. And what happens is that eventually, what happens is that the synthesis is also bidirectional, and eventually, these replication bubbles they meet halfway, and the two strands are separated from each other. Notice that you also have one old strand and one new strand. So DNA replication in eukaryotic cells and in human cells can be finished in a short time. Now the problem is whether we're talking about bacterial DNA or we're talking about eukaryotic DNA or human DNA. What happens is that the chromosomes become tangled up. Okay, they become tangled up, and they must be separated from each other. And that's why we need the other topoisomerase, which is known as DNA topoisomerase 2, and it's also known as gyrase in bacteria. It requires ATP, which is different than topoisomerase 1. Topoisomerase 1 does not need ATP. That's one difference. Another difference is that DNA topoisomerase 1 creates a single cut in one of the strands, not both of them. On the other hand, DNA topoisomerase 2 what it does is that it creates two cuts on the same strand. But eventually what it does is that it separates the two strands from each other. Now, because DNA topoisomerase 2 is important for DNA replication, for DNA division, inhibiting topoisomerases, whether one or two, can prevent cells from dividing as a result of blocking DNA replication. And as a result, topoisomerase inhibitors are used in the treatment of cancer to stop DNA synthesis and cell growth and proliferation. Now there's another protein that is needed, and it's known as PCNA, or proliferating cell nuclear antigen. And this protein, what it does is that it guides the DNA polymerases to the primers. Okay, so it's needed for DNA replication, and PCNA is used in diagnosing cancer, so it is a marker of cancer. Okay, why is that? Because cancer cells tend to do a lot of DNA replication and cell division, and as a result, they increase the synthesis of PCNA. And this is shown here by a technique known as immunohistochemistry. So this is done on tissue sections. Okay, so right here, these are normal cells, and what you see staining in brown right here, this is staining of PCNA, and you can see the brown color is a lot in these cancer cells versus normal cells. Okay. So DNA polymerases in eukaryotes are multiple, like in bacteria, but there are more of them. Okay, but I'll focus on 4 of these DNA polymerases, and these are DNA polymerase alpha, delta, epsilon, and gamma. Starting with DNA polymerase gamma, it's a mitochondrial DNA polymerase. Okay, it does have an exonuclease activity, so it is accurate, which is also known as the proofreading mechanism. Okay, now we have DNA polymerase alpha, delta, and epsilon. Now, alpha it has a primase associated with it. It has high fidelity, so it doesn't create that many mistakes. And probably that's because it has high processivity. That is meaning that it is a slow enzyme, so it doesn't make that many mistakes. On the other hand, DNA polymerases delta and epsilon both have high processivity, they're fast, they have high fidelity, they do not create that many mistakes, and probably because they have this proofreading mechanism, they have 3' to 5' exonuclease activity. So what happens is the following. So specifically, DNA polymerase epsilon is responsible for synthesizing the leading strand. Okay, now DNA polymerase alpha, as I said, is associated with a DNA primase. So the primase creates the RNA primer, and the polymerase starts synthesizing the Okazaki fragment. Okay, the beginning of the Okazaki fragment, but then it falls off. Okay, and DNA polymerase delta jumps on DNA, and it continues with the DNA synthesis. Okay. Now, both DNA polymerases, epsilon and delta, are associated with PCNA, so they are guided by this protein. Now, what happens is that DNA polymerase delta, as it synthesizes the Okazaki fragment, it hits the primer in the next Okazaki fragment, and it needs special enzymes to remove this primer, so it does not do this by itself, it needs help. Okay, and then but it fills in the gap created by these enzymes. Okay, so that's the idea. We have polymerase epsilon, DNA polymerase epsilon is responsible for synthesizing the leading strand. Each Okazaki fragment requires DNA polymerase alpha followed by DNA polymerase delta. Now the thing with eukaryotic DNA is that we have a problem with chromatin. DNA polymerase cannot synthesize DNA until DNA is free of histones. And what happens is that these histones are removed by proteins known as chromatin remodeling proteins. So they remove histones so that DNA polymerase can continue with the DNA synthesis. Now the same old histones as well as new histones can be used to refold DNA in behind the DNA polymerases, and this is done by chromatin assembly factors. Now, but we have a problem with the lagging strand, and the problem is that whereas in the leading strand, DNA synthesis continues until it gets to the end of the chromosome or DNA, we have a problem at the end right here where we have synthesis of the Okazaki fragment, but in order to complete DNA synthesis, we need to place a primer, but there is no space to add a primer. So the end of the lagging strand is not really completed. Okay, so this is okay. So right here, what happens again is that when you have this right here, and it's not completed, and you have another round of DNA replication, um, when this is removed, this becomes actually shorter. So one of the chromosomes becomes shorter right here. Okay? Now the thing is, the end of the chromosome is really important to, which is known as the telomere, is really important for stabilizing DNA. Okay? And this is completed, the lagging strand is completed by an enzyme known as a telomerase. Okay, and this telomerase is known as a ribonucleoprotein, meaning that it is a protein that is made of protein plus RNA, and that's why it's known as a ribonucleoprotein. Now, what is this RNA? It functions as a template. Okay, so it functions as a template. So what the telomerase does is that the RNA itself can hybridize to the end of the DNA. Okay, and what happens is that the telomerase elongates the template of the lagging strand. Okay, so it continues with the DNA synthesis. It makes it longer. Okay, it keeps on adding the same repeat, the same sequences, and that's why telomeres, telom by the way means end. Okay, so telomeres are composed of repetitive sequences of DNA. Okay, so this is what it actually does. It keeps on adding. It uses the RNA that is associated with it to elongate the template of the lagging strand. Okay, it keeps on adding the same exact sequence over and over again. Okay, so let's look at this animation right here. Notice that DNA synthesis continues until it gets to the end um, of the template of the leading strand. Now the other thing is that with the lagging strand, with the template of the lagging strand, this is the primer, but when it's removed, there's no space to fill in this gap right here. Okay. Now the telomerase comes in, and what it does is that it uses its the RNA that is associated with it, and it elongates the strand. That is the template of the lagging strand. Pay attention to something here. There are two mistakes in this animation. Okay, I want you to find out what these two mistakes are. One of the mistakes is in the type of DNA polymerase that synthesizes DNA, and the other mistake is with the RNA molecule that is associated with the telomerase. Okay, so why are telomeres important? Telomeres stabilize chromosomes. So when they are removed, when chromosomes become shorter and shorter and shorter with every round of DNA replication, chromosomes become unstable. And that leads to accumulation of mutations in DNA, and that leads to cell death. And this is really how we age. Okay, so remember this sheep right here? Her known is was Dolly. Uh, when Dolly was cloned from another sheep. The thing is that sheep, let's say that they live for 10 years and then they die. Well, Dolly lived only for 5 years and she died. And she died because of old age. Okay? and she had age-related diseases. Okay, and why is that? Because the DNA that was used to clone Dolly was 5 years old. That is, it was taken from a sheep that was 5 years old. So even though Dolly was 1 day old when she was born, her DNA was 5 years old, and that's why she aged quickly and she died early on as well. So this is the